my pleasure to introduce tonight's NYUAD Institute lecturer, my New York colleague, Professor James Canary, Professor of Chemistry at New York University in New York and the Chair of the Chemistry Department on the square, as we say, when we need to distinguish NYU New York from the NYU global sites. Professor Canary grew up in Reno, Nevada, in a somewhat different desert environment than one we have here in Abu Dhabi. A little cooler, a lot drier. He earned a Bachelor of Science degree in chemistry in 1982 from the University of California, Berkeley, and a PhD in 1988 from the University of California, Los Angeles, working with Donald J. Cram, a 1987 Nobel laureate who is credited with being a co-founder of the field of host-guest chemistry. After postdoctoral work with Ron Breslow at Columbia University, he moved in 1991 from the Upper West Side of Manhattan to Washington Square, where he began his independent career at New York University. He was promoted to associate professor and tenured in 1997, then promoted to professor in 2002. Professor Canary is a fellow of the American Chemical Society and has organized numerous scientific sessions at ACS conferences, as well as serving as the chair of the upcoming 27th International Symposium on Child Discrimination in Boston in 2015. He sits on the scientific advisory boards of Cryptomedics, Quell Therapeutics, Nanomedics, and Marutsan International, and he has served as a consultant to the U.S. Department of Justice. Since 2007, he has served on the executive board of the Harlem Children's Society, an organization that promotes scientific research programs for children and youth of families living in under-resourced and underserved communities. He has received grants from the U.S. National Science Foundation, the National Institutes of Health, the Office of Naval Research, the Office of Air Force Scientific Research, and private organizations to support his research. His research interests are in synthetic chemistry and molecular chirality, which is the idea of handedness or asymmetry in molecular structures. That is, sometimes molecular structures differ from each other in ways that are akin to the difference between your left and your right hand. They are non-superimposable mirror images of each other, and this bequeaths them with different properties. For example, try shaking the left hand of your neighbor with your right hand. It's awkward compared to a right hand to right hand handshake. Similarly, the handedness or chirality of molecules in nature has a profound impact on the way some molecules interact with their environment. Professor Canary's research has led he and his co-workers to exploiting the chirality of molecules to design and synthesize synthetic materials that can behave as sensors that can correspond to their environment and switches that can manipulate their environment. Tonight he's going to tell us about the single-handed molecules of life. Please welcome Professor James Canary.
Thank you, Tim, for that generous introduction. It's really fun to be here this week and to experience the community that you have in Abu Dhabi, and especially NYU Abu Dhabi. I'm so impressed with everything that's going on. And I'm very happy to talk to you a little about the single-handed molecules of life, which is a lifelong love of mine. I'm going to begin my talk showing you just some familiar objects, and Tim already alluded to some that display handedness, and then talk about how different molecules can have the property of handedness, and briefly mention some materials, and then how does handedness of molecules affect health and life. Now, I want to use a new term. Many of you are familiar with chiral, which is actually from the Greek word for handed, and it means asymmetric in such a way that its mirror image is not superimposable. And Tim already used the example of hand. If you're still paying attention, you'll notice that this particular example, there are two hands, but they really are the same hand, aren't they? This is just the front and the back of the same hand. And so those two images can be superimposed on one another simply by rotating one of them and mapping it onto the other. But really, it's the left and right hand that are mirror image. And no matter how you try, they don't match up. And so we're going to be doing this with different examples through the talk. Of course, you have you're very familiar with many of these examples. For example, screws are chiral. They have a handedness, and by convention, the screws that we buy normally are right-handed, so that when we turn our screwdriver clockwise, they go into the wood or the sheet metal. We also have drill bits. All again, by convention, are right-handed. Many different shapes and sizes. This one for digging very large holes in the ground. Flowers can exhibit a chirality, a handedness. If we look closely, it's a little bit hard to see in this image, but the edges are overlapping in these flower structures. This is one flower that I first encountered in the New York Botanical Garden. This one is a desert lily taken in Nevada, my home state, in the desert. Of course, it only blooms for about 3 days out of the year. This one, I don't know what kind of flower it is, but it also has this propeller shape. I took this picture this morning out on the grounds. It's a very lovely flower. You can tell me later if you know what it's called. But all of these have the shape of a propeller, and the propellers have handedness also. For example, in a boat, if it's a right-handed propeller, then if you turn the blades in the direction of your fingers, and if it moves in the direction of your thumb, then that's the handedness. So right-handed propeller, you turn it this way, it goes to my left, your right, left-handed the other way. We observe chirality in animals. A familiar example is snails. Their shells can follow a handedness rule. It's very interesting. These up here are actually two species. On the left is left-handed, is Neptunia angulata, and the right hand is Neptunia dyspecta, and they have nearly mirror image morphologies. Other snail species can have either right or left-handed morphology. We see handedness in art. This is, of course, Michelangelo with Adam and God and the Sistine Chapel. And we have God with his right hand pointing at Adam, who's holding out his left hand. And it's partly beautiful because there's sort of a symmetry there. Sometimes we like symmetry is beautiful. This is Rodin's Cathedral. This is one of my favorite pieces of art, but in this case, there are two right hands. And it forms a beautiful helical structure, like a spiral staircase or like DNA that we'll talk about later. And it evokes a sense of cooperation together because two different people would be required to show that image. Our faces are almost symmetric, but not quite. Often, a more symmetrical face is considered more beautiful. The newscasters tend to have very symmetrical faces. Don't look at mine. This is a cover of a journal. Later, I'll tell you that we work on electronic control of helical chirality, and the editors thought it was very funny. It would be very funny since my name is Canary, and since my article is about inverting the chirality of molecules to show a canary looking in the mirror. Two-dimensional structures can be chiral. This is the floor of Boeb's library at NYU, New York, and it's modeled after the floor of San Giorgio Maggiore Church in Venice. When I was hired at NYU many years ago, it was thought that this three-dimensional aspect that the floor conveys prevented people from jumping and committing suicide in the library. But unfortunately, that didn't hold true, and now there are barriers. But I wanted to show you what you can do. If we place the same image side by side, repeating it, and if we just flip the right one, then we get to the enantiomeric image of the left one, and it becomes symmetrical, and it looks beautiful if we compare again. But that works in 2 dimensions. Handedness in art and in other venues can communicate motion, and this is the Central Park, New York merry-go-round. So I don't know why chirality communicates motion, but maybe it's this is one reason, because we're used to, the merry-go-round in this case goes around in a counterclockwise direction, and so it has a asymmetry associated with it. This is the helix of the entrance to the Lincoln Tunnel, which is in New York. It goes under the Hudson River. And it has a particular handedness. It would have a non-superimposable mirror image. And again, with the cars, it has the sense of motion in a helix. So in this artwork, which is, of course, Van Gogh's Starry Night, don't the swirls give you the sense of the wind blowing in the air? And so the curvature and the chirality communicates motion. This is a piece of artwork called City of Industry. It's in the Chanin Building, which is across the street from Grand Central Station in New York. There are a series of relief images like this. This is sort of the thinker, and he's sitting in a spiral environment, and it's meant to convey industry, that is activity, the city in action and all that goes on. And this chirality, this handedness, communicates that through this art form. This is actually made by René Paul Chambellan, who studied for a time at NYU, New York. Of course, here is the famous Guggenheim Museum in New York. It's the old Guggenheim, and the spiral walkway on which most of the art is shown on the walls. Of course, you in Abu Dhabi are taking dissymmetry to new heights with your coming Guggenheim incarnation. So anyway, these are all examples of chirality or handedness in different areas. And what about molecules? I'm going to talk about, just ever so briefly, very different examples. And all of these molecules, and DNA and proteins, of course, are some of the key molecules involved in living organisms. And we're going to see it all comes down to carbon. Everything is derived from the element carbon, which is this C here. And carbon has a tetrahedral geometry, and it can have. And it can be attached to different things. And when it's attached to 4 different things, then it turns out it has mirror images that are non-superimposable. And in fact, we have a whole nomenclature that we can use in organic chemistry to assign, systematically whether something is right or left-handed, and you can use your actual hands to make this definition. But we won't go into the details of it. So I want to start with DNA because it, I think, is one of the most familiar chiral molecules to most people. And this is the most common form that people see, and it's a right-handed helix. So if you look at it as a screw and you turn it in the direction that it would go forward like a propeller, or it would screw into the ground, you can easily see that it's right-handed. There's another form. This is the B form. This is the form that typically is adopted by RNA, the A form. Over here, it's also right-handed, by the way. This one is the Z form, and it doesn't occur very often in biology, definitely not very often. But it actually is left-handed, and it's a little hard to see, but it has a bit of a zigzag structure. So where it's starting out here, going behind, and then coming out, and then behind. And so if we turn this one, it has a left-handed helical structure. So it's very interesting that you can have left-handed DNA, but this only exists with special sequences of the base pairs and under special conditions. So it's not very common. These two forms are very common. How are they different, though? They have a similar pitch. That's the distance between, across one turn, and other somewhat similar characteristics. But DNA, you can think of the A form of DNA if you started out with it flat, with the two backbones running along each edge of this rectangle, and then just twisting it, you would get the B form, which is the one that was in the middle on the previous slide. However, if you wrap this around a central axis, you get the RNA, or the A form of the DNA. So here they are again. This is the one that you just twist, and this one you can see how it's something sort of flat that's wrapped around a central object. So really the DNA, it ends up being essentially a crystal, because these organic units that are running across are close together, and it's like a one-dimensional repeating structure. But the edges of the chain don't match the distances in the middle. And the only way for the thing to wrap up in a double helix like this is for the outside to twist. So it's a distortion, because the distance between the sugars on the backbone is different than the distance between the bases that are stacking. So this right-handed, left-handed business was used by Ned Seaman in the chemistry department in New York to create a device, because choosing a particular sequence in the middle of this DNA construct and then attaching other DNA moieties and then placing dyes in the proper position, this yellow part can be interconverted from right-handed, as shown here, to left-handed, as shown here. It's done just by increasing the concentration of ions in the solution. And so only this yellow part is susceptible to this inversion. And the remainder has a different sequence of nucleotide bases, and they remain relatively unchanged. But as a result of this twisting from right to left, the distance between two interacting organic moieties changes, and this can be read out using a particular type of spectroscopy. And more sophisticated devices have been developed since then. So so nearly all the naturally occurring DNA and RNA is right-handed helix. And what makes it right-handed? So here is a detail of the structure of one of these units. This thing with the N's and the O's is one of the sort of blue guys here, and this is a different kind of model. This is a space-filling model, and this is just a stick model. And then the sugar is in this. This is a monosaccharide. It's a fructose. It's a ribose, which is part of this region here. And in the ribose structure, there are 3 carbons that are attached to 4 different things, and so they can have mirror images. So this building block of DNA contains carbon atoms that display handedness so that that handedness dictates a right-handed helical turn in DNA. Not only do we find helices in DNA, but we also find them in proteins. So in proteins, there are different types of structure. Shown here is the sequence of amino acids in a protein, and they're just single-letter abbreviations. It's not so important what they all stand for, but each one of these letters represents a monomeric building block in a molecule that, when the monomers are joined together, forms a protein. These fold into different possible structures, and one is a helical structure known as the alpha helix, and another is a beta strand. But the secondary or larger chirality is associated with the helices. These then join together in a longer protein sequence. One long linear molecule like this one may have regions like the red part that become helices and regions like the blue part that become beta strands. And then these may fold into a three-dimensional structure and then can go on to form even greater levels of structural organization. But it's this helix that is kind of intriguing for my talk today. In this more detail, we have a sequence of 9 amino acids forming a right-handed helical structure. Again, using our drill bit illustration, we can see it's right-handed. And then if we include more atomic detail in the sequence, we can see that this structure comes down to carbon atoms again. For example, this one. This is a carbon atom attached to 4 different things. It's another carbon here attached to an oxygen atom, attached to a smaller one, attached to a blue one, and then this R2 label. So there's 4 different things. So that's a chiral atom, and it's the handedness of that atom that biases the conformation of the helix to form a right-handed structure. So whenever we talk about chirality, we have to, there's a few people that really made fundamental contributions, and I'm sure you are familiar with Louis Pasteur, pasteurization, and also he made really fundamental contribution to this discovery that molecules could have handedness, that they do have. And it was a very special set of circumstances that led him to make this statement, which was really transformative in chemistry way back in a couple hundred years ago. I don't remember when it was. But so what he observed was with tartaric acid. And I've begun to draw atoms in the molecules. But he didn't know, just like some of you, he had no idea about this part. He made crystals of this stuff. And it turns out that this substance, tartaric acid, can form crystals that have non-superimposable mirror images. And he saw that just by looking at the little crystals under a microscope and just noting it. And then and then on the next slide, I'll show you what he did. This is something that is somewhat rare. And it's actually, those of you who follow the chemical structures, it's not exactly just the tartrate that was used. It was a certain salt. So molecules can crystallize in different forms. In this particular molecule, the 2 mirror images crystallize in 2 different crystals. So all of the left-handed molecules end up together in one crystal, and all of the right-handed molecules end up in a different crystal. That's called a conglomerate. And it's a little bit less common than other types of crystals that are formed. And this is just a diagram showing the relationship between the amount of, let's say the left-handed one is called D and the right-handed one is called L. So it's most highly crystalline, and it melts at the highest temperature at 100% D or 100% L. And at the 50-50 point, it's the lowest melting. So that means the molecules, when they crystallize in this form, are most stable. And so that's what they prefer to do, to give you right-handed, left-handed crystals. An alternative form is a racemate, where, in fact, a one-to-one mixture of right and left-handed molecules in the same crystal is the most stable situation. So Pasteur was lucky because this is a little bit unusual for a substance to form a conglomerate. And normally, tartrate and derivatives of tartrate form racemates. It's just a particular one he happened to have studied, and that led him to think about, well, what could this mean? Why would I have two different crystals that have non-superimposable mirror images? So what he did is to look at them under the microscope and use tweezers, and one by one separate the one form of crystal from the other. If he then dissolved the crystals, he would observe something called optical activity, which I'll explain in a moment. But one crystal, all of the crystals with one shape gave the same optical activity, and all of the crystals with the other shape gave the opposite optical activity. But if he dissolved the mixture of crystals, there's no optical activity at all. So what is this optical activity? It's simply the ability to rotate plane polarized light. And in fact, you know what plane polarized light is because you may have at some time had Polaroid sunglasses, which have basically lines across the glass very closely spaced. Light in its native form has waves propagating in all different possible directions. But on passing through such a glass, only the waves that line up with the narrow lines are allowed through. And this reduces the amount of light that passes through, and so it can protect your eyes. Also, some of the 3D movie technology now uses this type of polarization to give you a stereographic image. But anyway, most people have some familiarity with polarization and linear polarized light. If we take the light that's been polarized in this vertical direction and then use another pair of glasses that have the same orientation of lines but rotated 90 degrees so that the polarizations are going this way, then no light will pass through at all. So it's this plane polarized light that's introduced to a solution, and we can determine if the light waves have been rotated by the substance in the solution. So this is a unique property of chiral molecules when they're dissolved. If you have an excess of just one of the mirror images, you are likely to see this phenomenon of optical activity or rotation of plane polarized light. If you have equal amounts of the opposite-handed molecules that dissolve, then the right-handed ones rotate the light in one direction, the left-handed ones rotate it in the other direction, and it cancels out, so you see nothing. So this was a key observation. Other folks were involved with molecular chirality as well. Wislicenus studied lactic acid, which is what causes you to feel stiff after you've been running. It's produced especially in your muscle when you don't have enough oxygen in your blood. Of course, he didn't know these molecular structures either. Other guys joined in the discussion to speculate that they had some idea what elements were contained in the lactic acid. And so these were the kinds of structures that they drew. So these two, you can see, are two tetrahedra with 4 different things attached and are mirror images. And they cannot be superimposed, this one on that one. So what Licinus's observation was, is that from muscle you obtained a lactic acid that rotated light and was chiral. But if you were to obtain it from another source, such as chemical synthesis, it did not have this property. It did not rotate. It did not have optical activity. And so Van Hoff came along and came up with the notion of carbon having a tetrahedral structure and having this property of chirality because of the non-superimposability of the mirror images. So this was some history from 1873. Well, now I want to, I've gone into a lot of detail, so I want to go back again to other things. So these are crystals of different sorts, and Panch Nomov here would know much more about this than me, in fact. But this is from the lab of Bart Carr, my colleague at NYU New York, showing different crystals on different size scales from 5 millimeters, 1 millimeter, 100 micrometers, which is 10 of them, it's 1/1,000,000th of a meter, that are all twisted. And many of these have been studied. Here's one example. He published a paper last year studying aspirin that forms twisted crystals. So here's a scanning electron microscope image. And if you look here, there are long fibers in these aspirin crystals. And if you look in this region, they look like they're narrow. And in this region, they look sort of flat. And then they become narrow again. So what we're seeing is actually we're looking edge on to a helix, where we see the narrow edge in this region, and we see the flat part in this region. So it's a helical structure. But there's no way that aspirin itself, all by itself, would form a helical structure. And in fact, all of these twisted crystals, this is not a property of the molecule itself. They should pack into shapes that would not give twisted structures. And what Bart and his colleagues determined was that the twistedness comes from impurity of salicylic acid that's present in the aspirin. And so the degree of twistedness depends on the amount of the impurity that's present. So the impurity doesn't pack together as well as the aspirin, and so it causes a dislocation. Remember, just like I said about DNA, where the spacings between the bases that are stacked in the middle are different than the spacings of the sugars on the edge, and that causes the twist. Well, the same thing happens here, where an impurity causes a dislocation and results in a twist, and in all those twisted structures. Now, Bart and Mike Ward in our department recently published a paper with these images taken by atomic force microscopy. This one shows a spiral structure that is chiral, isn't it? It would have a mirror image that's non-superimposable. These are images of L-cysteine, which is something that forms a certain type of kidney stones. That's a real problem. And the nature of the research is to understand how these kidney stones form and to find ways to prevent them from forming, because this is a very unpleasant and sometimes very serious health problem. This is another structure, though. This one on the right, it's a zoom in. This is a 1/1,000,000th meter scale. This is less than that. This is 5 times larger scale, smaller, so zoom in 5 times. Nanometers. Well, I knew nanometer, so these talks are very funny because you try to be a general audience, but then you have the provost here. So this one is not spiral, is it? This just forms a hexagonal ring. And so anyway, this atomic force microscopy imaging is allowing them to sort out fundamentals not only of how the molecules are packed together in 3 dimensions in a solid, but how they go from a solution to a solid, and to figure out then strategies for preventing that from happening. Another example of a material that has chirality is this polymeric structure. This is actually from the laboratory of Mark Green, who's a professor in the NYU School of Engineering, which is a brand new school since NYU New York merged with Polytechnic University. But this is the structure of this backbone here, and you can see that it forms a left and right-handed helices. This goes down and then up and down, and so it's mirror image helical structures. Whether you have a right or a left-handed helix depends on the nature of the attachment. And what's not shown here, this little thing is a bond, so there would be another couple of atoms attached here. And what Mark's great contribution has been, of course, if all of the atoms attached to this helix backbone had chirality, as is the case with DNA and proteins, then it would bias one form or another, depending on the arrangement of the attached atoms in 3-dimensional space. But what he found is you don't have to have all of the groups exhibit molecular chirality. You can only have a fraction. In fact, if you plot the percentage of side chains that have handedness or single-handedness or chirality, it's nonlinear. So it only takes a few percent to get a very strong induction of one helical form or another. And so he describes this as if the side chains that have chirality are sergeants, and the side chains that do not have chirality are soldiers, and the soldiers are doing what the sergeants tell them to do. And this is a very simple but profound description, because it turns up in many different phenomena. And there are many papers that cite his work today. This is one example from my laboratory. I'll be talking about this tomorrow to the chemistry department. But we work on molecules that bind metal ions. So this is a structure. There's a copper metal is in the center. And this part has no copper. These are, N is nitrogen, and the other things are all carbon in this structure. And so the gray and the blue are this one shown here in 3D. And when you present the copper ion to this, without any copper, it's flat or it's floppy. It has no defined geometry. But on forming a complex with the copper, this wraps around, forming a propeller-like structure. And that propeller can be right or left-handed. And so we have shown that we can interconvert right and left-handed structures by adding one electron or reducing one electron. So it's almost like an electrical switch on the wall, but instead of the light coming on and off, we're turning the glove inside out from right hand to left hand. And so we're interested in pursuing this in some applications involving polymer chemistry that I can talk about tomorrow. But the recent example that we published, we used this molecular switch chemistry to demonstrate that we could synthesize, we could use it as a catalyst or as a modulator of an organic reaction where A and B combine to form some product. And if we use, say, the right-handed form, we get a right-handed product, but we could use an electron to convert it to the left-handed form and then get the left-handed product. So this is a really exciting fundamental stereochemistry and also has some interesting impacts for making things like drugs. And we'll talk about chirality of drugs soon. We also have used the same type of copper adding and removing electron chemistry with peptides to convert them. Remember, I told you about the alpha helix of peptides and beta sheet. So this chemistry interconverts from alpha helix to beta sheet form. We just published that a couple of years ago. So at this point, I've been persecuting with molecules. Let's review the terms, class. We have learned chirality, which means handedness. I'm using this word enantiomer, which refers to one of the two possible mirror images. So you have a left-handed enantiomer or a right-handed enantiomer. And a racemate is a one-to-one mixture of enantiomers. And we'll probably use those as we're describing some of this. So now we want to talk about how does this impact health. And I'll give you couple of cherry-pick examples, just showing molecular recognition between molecules, proteins, drugs that are being developed at NYU, and then drugs that you are buying at the pharmacy. So here's one example. This is cholesterol. We all are familiar with cholesterol. And it only exists in nature in one enantiomer form. And it's this one right here. It has all of these dark wedges mean that there's an atom coming towards us from the plane of the wall. And so there are 1, 2, 3, 4 that are obvious. And then there are additional chiral centers that are present here, here, here. All carbons with 4 different things attached. But the mirror image of this, which would be here, has not been found in nature. So this is only made in our bodies, sometimes to a greater extent than we would like, in a single enantiomeric form. And it turns out that it's made starting from essentially vinegar, which is a 2-carbon molecule, which is transformed to a long chain of carbon atoms. This is squalene, which is in shark liver oil. And there's a catalyst or a modulator for the reaction that binds to this and folds it into a particular shape. And that shape is just right so that when bonds are formed to convert this long linear molecule into this one that has multiple rings, that you only get one enantiomer. So there's some molecule in nature that mediates this reaction so that only one enantiomer product is formed, and it must be chiral. Here's some other examples of molecules that we care about. These are amino acids. So this NH2 is an amine, and this group here with the OH and this double bond O, that's the acid, so hence amino acid. Different amino acids, there are 20 common ones. They have different side chains. So this is asparagine, and it was long ago found to have a sweet taste. But so L-asparagine is the natural occurring enantiomer. The opposite one that we can make in the laboratory tastes awful. So we must have receptors in our tongue that have this property of chirality that recognize asparagine the way we would like to recognize it and not in some other way. This is MSG. So Chinese food has a lot of this, and it tastes good in the natural L form, but the enantiomeric MSG has no taste at all. And this is aspartame, which is a sweetener. It is NutraSweet, which has a couple of these amino acids, and only the ones that have the correct chirality have the sweet taste. Also smells. Okay, so here is a structure. Again, it's not at all critical what the lines mean, but you can easily see that these are mirror images as I've drawn them. But one enantiomer smells to us like spearmint odor, the other like caraway. In many animals, but especially insects, the smell is associated with attraction of the mate. And if you're a little insect in the field, you have to find ways of finding the girl or the boy insects. So they are very sensitive to these smells. And some, like the German cockroach, it's called a pheromone, the sex pheromone. It has 2 of these carbon chiral centers, but it does not care at all which mirror image is present. It responds to all of them. However, the tsetse fly responds to this long molecule, and it has 2 chiral centers, but this is the only form that has any activity at all. If you have the mirror, if you have the other one of this carbon center where instead of the wedge coming up, it's going behind, doesn't recognize that at all. The olive fruit fly, the male responds to one mirror image and the female to the other one. And then in some cases, different species respond to different enantiomers. So it's very interesting. And so some of these are used in pest control, and it's important to be able to make such compounds in the correct enantiomeric form and in the right purity if you want to be able to use these to confuse the insects so they can't find their mates. So here's another example of recognition between molecules. Work going on in Bobby Aurora's lab at NYU, New York. So here he's interested in, these are the proteins that have the 3D folded structure I showed you earlier. And he's interested in proteins that recognize each other, and our biologists here are all very interested in this kind of issue at one level or another. And so his lab identifies such recognition events in biochemistry, and then they try to develop mimics that can block one protein associating with the other. And then they use this to target different diseases or different biological phenomena. In chemistry, we are often focused. No, I'm not going to say that. We are interested in just biology, but we're also interested in medicine because it's closer to where the money is. So no offense, I'm getting myself in trouble. So Aurora Lab has developed sophisticated ways of mimicking the alpha helix. So here's a natural alpha helix depiction. And so they, in one strategy, replace a weak interaction in the alpha helix with a solid link. So here the dotted line means that there's not a real chemical bond. There's just a very small interaction between 2 atoms that cause this helix to fold. And they replace that with a solid line, which locks it in and makes it a very effective mimic of the original helix without perturbing the other parts of the structure. And it also stabilizes the molecule against attack by protective mechanisms that the cell has. So this can be a drug. And there's other strategies where they just use a backbone that roughly resembles a helix backbone and then attach groups at positions to mimic the location of positions in a naturally folded alpha helix. And they've used this recently to demonstrate that they can affect hypoxia-induced signaling. Hypoxia is a low oxygen concentration. It's very important in a lot of biological phenomena. And if you have tissues that are oxygen deprived, there are responses by your natural proteins that then cause signals the cell interprets in different ways. And so for health reasons, we're very interested in being able to control that, and especially in cancer. Cancer is uncontrolled cell growth. Tumors typically are very low in oxygen concentration because the cells are going fast. They suck up all the oxygen, so they're oxygen deprived. So that's hypoxia. And so this could be used to kill the tumor by blocking the cellular response to that low oxygen. And so these different strategies have been effective. You can see a mouse with a tumor treated with their drug has lost the tumor, in this case, a reduced tumor size. So it's very exciting. But let's talk about drugs you're more familiar with, ones that are on the market. Of the 30 top-selling drugs on the market, 14 of them are proteins. 16 are organic molecules that, like what I've been talking about. 12 of them have non-superimposable mirror images. All of these, of course, have non-superimposable mirror images. So the vast majority of drugs on the market have this property of chirality. And there's some interesting aspects to it. Because it turns out if you have the racemate or the right-handed mirror image or the left-handed one, these all may have different activity, because I think we've shown through these other examples that your bioreceptors have this chirality feature. And so if you have one mirror image or the other, you're going to have a different interaction between the molecules. And if you have both, you may have two sets of reactions. So this is important for bioactivity and also for some monetary reasons that I'll mention. So a classic one to talk about in this case is thalidomide, which first was prepared in 1953, and it was never approved in the US by the FDA. And I'm sure they are very glad they never did, because it was used outside the US and was found to be to cause birth defects. And this word teratogen is Greek for monster. And so the children would be born with limbs that were way too small or missing, and it's just terrible. And it's customary to show these awful pictures, but my host, Professor Dorr, his wife is about to have babies, and I thought it would be in poor taste to show birth defects. More recently, this drug has attracted attention again for treatment of cancer and other applications, and has now been approved, but with strict restrictions so that women will not be taking it if there's a chance that they may be pregnant or may become pregnant. But it has such good activity that it has now come back again despite this awful record. Well, what about this issue? It turns out that one enantiomer, one mirror image of thalidomide is not teratogenic, does not cause birth defects. The other one does. However, if you take either one of these into the body, they both cause the birth defects, okay? So how could that be? And it turns out in the body, the two mirror images are interconverted between each other. It's a special property that is unfortunate in this case, because It means you cannot just isolate one mirror image and use that to avoid the teratogenicity. But all of this took a long time to sort out. A more contemporary example is this one here. This is omeprazole. And here I've shown it as the one-to-one mixture of mirror images, so it's the racemate. That was sold by AstraZeneca and called Prilosec, and used to treat gastroesophageal inflammation and esophagitis and ulcers. In 2000, it was making the company $6 billion a year. That's amazing drug. But their patents, the key patents, were due to expire in 2001. So what happens when the patents expire is that the companies that make generic forms of the drug can now jump in and make it and sell it. And so this has a huge impact on the company's profitability. So what they did at this time was, well, they went back and they decided, let's develop a better form of omeprazole. And they looked at quite a number of different molecules, but the one that they found was the best was just the single enantiomer, esomeprazole, which is now sold as Nexium. And I know my mother cannot live without Nexium with her ulcer, and many people benefit from it today. But it's just simply the single enantiomer of what was previously sold as a racemic mixture. And so this could be covered by new patents. And it was approved by FDA, and it continues to be a multi-billion-dollar drug today. So this is called a chiral switch, where there's a drug that was previously sold as a racemate and then developed and marketed as a single enantiomer. And there's evidence from the company that says that this is more effective than the earlier drug. However, this is not a phenomenon that's without controversy. Because, you know, in the racemate, it was probably that same enantiomer that had the activity. And so some folks say, you know, hey, this is kind of cheating, because there's a reason we have a limit on patents, so that drugs can become more affordable. And so this study looked at 9 examples. We call these chiral switches. Sorry, this is 2000 to 2011. That's a typo. And the key issue is that the approval of the new single enantiomer drug does not require clinical comparison with the old drug. You only have to show that it's active by comparing it against a placebo. And that's all the regulation is in the US for getting a new patent and being able to market the drug. So these folks argued that these companies need to be required to compare the single enantiomer with the racemic drugs, because it may be that some people are paying big bucks. In fact, my mother cannot afford to buy the Nexium. She sends a letter to the pharmaceutical company every couple of months, and they give her a free supply. And that's the only way she can afford it. She's on Medicare. Of course, now I probably seem like an awful guy. Why don't I just buy it for her? I hadn't thought about that. But anyway. So today, though, the new drugs coming out, the FDA in the US really wants to see the activity studies done on each enantiomer and on the racemate right from the beginning. So I guess this chiral switch thing is going to become less of an issue. All right, so what does this have to do with life? And obviously health is related to life, but maybe this is a little bit different set of issues. I want to, if we have time, talk about how does life produce this issues. It turns out all the biological molecules that are handed, they all have the same handedness. They're all right-handed. So all the amino acids are L, and all of the sugars are D. And so they all have the same rough three-dimensional structure. And how did that come about? What was the origin of that? And then is this issue of the life's molecules all having the same handedness, which we could call homochirality, homo meaning the same and chirality meaning handedness, is that actually just tied in with our whole concept of life? So this is how does a snail shell become right-handed or left-handed? And they don't know. Okay, but this was an experiment aimed to try and discern this. This is by Reiko Kuroda, who's one of my heroes. She's a female chemist in Japan, who's nearing 70, and so she's had a career in a country that hasn't been welcoming to her, but she's done very well. So this is one of the things she has studied, and in the development of the snail, she found if she just introduces a probe and distorts the cells in a certain way at a certain time of the development, that she can switch a snail that would naturally have, say, a left-handed shell to one that has a right-handed shell. So it's kind of a crude thing. It's nowhere near as sophisticated as an experiment done by Claude Desplan many years ago. And there are lots of people here who know more about this than me. But he studies flies, and especially vision in fruit flies. And if you look here, this is several receptors in the fly eye. Of course, the fly eye has this multi-component cell. So this forms the ommatidium, and these two are mirror image, two-dimensional mirror image structures, because this one kind of goes up more on this side than that side, and this one goes down. So it's like there's a mirror plane right here. And as they're arranged in the eye, all of them with the same planar chirality are on one side of this line, and those with the opposite are on the other side. And so what this paper describes is this was developing a new technique to get these kinds of images so that you could actually see this phenomenon, and then you could study the molecular biology that results in this phenomenon of the mirror image eyes. And they could distort the symmetry by changing different molecular structures in the expression. And so there's been a lot of work, I noticed, in that area. Since then, I didn't see that they continue to study that particular phenomenon, but it's very interesting. So those are two examples of sort of how there's a whole field of how does the living thing have one chirality or another. And it does boil down to things that can be studied and probed and established in molecular biology and chemistry. So here is the question of how did we end up with all of the amino acids being L and not the opposite, let's say all right-handed, and all of the sugars being D and not L. And then how they come to dominate. Or, sorry, this is the question of why L not D, and this is the question of why are they all L and none of them are D, or hardly any. So the issue about why L and not D is, to me, a little less interesting, because there are many ways that you could wind up with one chirality or another. And it turns out that nuclear physics tells us the universe is not exactly racemic. And so it's very subtle and like the weak nuclear force, but it could be possible that some chemical phenomena could be influenced by particle physics asymmetry. Others have argued that molecules came from extraterrestrial sources. And I'll tell you one argument about that. It's not as fantastic as it sounds initially. Or a phenomenon called circularly polarized light, which could cause selection of one enantiomer or another. Or even some have talked about, you flush the toilet and the water spins one direction or the other direction. That one is not very likely. So here's one example from Ronald Breslow at Columbia, who claims this as a plausible approach. So his goal is just to show, well, here's one scenario that would work. Doesn't mean it was the right one. But it is well known that the Murchison meteorite had several amino acids that were all the same chirality. And here they are, okay? These are not the kind of amino acids we have in our proteins. These have a methyl group, a carbon. There's a carbon in this Me structure, so that this carbon that has 4 different things attached to it, and it's the center of chirality, is fixed into this particular configuration. And for example, when the meteorite lands and is very hot, it's more stable and maintains its chirality. And so all of these have very analogous structures, except this group here differs. And they all have the same handedness at this carbon center. But they only have an excess of a relatively small amount, 15% in the best case, of one enantiomer over the other. But he says this is a way that chirality certainly has come to the Earth. So then requires then, for his explanation, some way to transfer the chirality of these extraterrestrial amino acids to ones that are found in our own biology. And so he was able to demonstrate a reaction, and I won't trouble you with the details, but the efficiency of the transfer from the outer space amino acids to the Earth ones was very poor, only resulting in 1% excess of an enantiomer. But it was the right one. So those L amino acids from the meteorite induced the L amino acid in the Earth amino acids. So an amplification mechanism would be needed somehow. And so it turns out that Breslow and a very smart person, Donna Blackman at Scripps, proposed a solution in 2006. However, they were a little late to the game, because Herb Morowitz, who is another faculty member at NYU Engineering, although at that time it was Polytechnic University, had already explained this in 1969. And I don't think either one of them had that citation in their paper, but later it was pointed out to them. But anyway, and it goes back to the fact that amino acids form racemates that are less soluble than the single enantiomer. And this is the picture I showed you earlier when we were talking about crystals. And so we have at 50-50, D to L, you have the most stable structure. So what can happen, let's say you have a mixture of L and D, and I've shown here 7 red and 6 blue. If these form a racemic crystal, that means exactly 1-to-1 mixture will be removed from the solution and form a solid, leaving behind an excess of 1 enantiomer. And so this would be the way that you could end up with a solution that has a very high purity of a single mirror image, even though what you started with had a very close mixture. And so then this could go on and become part of an organism, however that happened. He also showed that these same, that amino acids, earth amino acids now, so let's assume we can make earth amino acids of the L variety. And for all of the ones studied except proline, they can catalyze a very basic reaction that results in the most simple sugar, which is glyceraldehyde. And this reaction, the catalyst is the amino acid. If it's L-amino acid, you get D-glyceraldehyde in excess. And it turns out, once again, that glyceraldehyde, as a mixture of mirror images, forms a totally insoluble solid in water, but the D-glyceraldehyde is very soluble. So there's the same amplification mechanism available so that you can get the D-glyceraldehyde, which is a starting point for making other sugars, including ribose. So see, glyceraldehyde has this part of ribose came from glyceraldehyde, and this chiral center is the one that makes it D. So as drawn here, the OH is on the right. And so this becomes part of the DNA. And so there are chemical mechanisms that can account for, starting with amino acid in a meteor hitting the Earth, resulting in L amino acids and D sugars. And so this is just a summary of what he has discussed. And this is work that's ongoing. There's still many questions. I'm sure you have several. What about, there's a lot of interest in, is there life elsewhere? And I think I've tried to show you today that chirality certainly is an inherent feature of any life that would be based on carbon. And there certainly are places in even our solar system where there is lots of carbon. For example, Titan, which is a moon of Saturn, has oceans of carbon. They're methane and ethane. They're very small hydrocarbons. They're not like the petroleum around here, but it's very cold. But deep underneath, it's warmer. There could be water there. It's possible that some kind of carbon-based life could exist there. That would be very interesting if we could find it. If it's there, and if it's based on carbon, then we should be able to detect it. I mean, how would we know? And one way would be to study those carbon compounds to see if we can find molecular chirality. How do you do that? One way would be using like the optical rotation, which is a form of spectroscopy. But we can't do that using telescopes. It's too many artifacts. It doesn't work. We could separate and analyze the carbon compounds on Titan, but that's very difficult. To send a satellite, the instruments that we use to do this are pretty sophisticated, and they have to operate in extreme environments. We could try to retrieve the carbon compounds and bring them back to the Earth to study, but contamination is an issue. So this is an unsolved problem. 2 days ago, NASA sent out a call for proposals asking who wants to work on problems like this. So maybe some students would like to take that up. So finally, if we have time, we're almost out of time. Let me briefly mention that at NYU, New York, we have work going on that shows self-replication of chiral molecules. And this is one of several aspects that are part of the definition of what is a living thing. And so Seaman, again, together with Paul Chaikin and Dave Pine, who are in the physics department at NYU, described an example where they self-replicate essentially a sequence of DNA. And I think I can just show it with this slide. If we start with a linear sequence that has two different, it's made up of cassettes, green cassettes and red cassettes. And these can assemble together only with red and green cassettes. So if we have a long line that's covalently bonded together and we introduce the building block cassettes, they will associate with their complementary, Legos, if you will. And these are Legos. You can not quite see it, but this illustration is Legos. So, and then link together to reproduce the same sequence of cassettes. And then these can dissociate again by clever procedures that Chaikin developed to produce two daughters. And each of these can then combine with the building blocks and reproduce themselves. And then those can be separated and thereby demonstrate self-replication. And I'm going to skip the details as we're about out of time. But it's a step towards showing really fundamental aspects of life. And we're going to see some important aspects of chirality come out of that work before long. So anyway, in my talk today, I would like to just summarize that chirality is both beautiful and it can be meaningful in our art forms and other places. But it's also an essential characteristic of life on Earth, the molecules that are involved in living things. The chirality can be measured, we can manipulate it in chemistry and biology to change chiral aspects of organisms or control properties of materials or improve our health. The origin of the biological homochirality certainly is not known, but we've begun to uncover some chemistry that could explain how it might come about. If we were to observe life elsewhere, we ought to observe this excess of single enantiomers. And so if we could figure out how to do that on other planets and moons, that would be really great. So in sum, chirality is central to life and life processes. And I'd like to thank Ned, Bobby, and Bart, who actually gave me some slides for this talk. And I also need to acknowledge the National Science Foundation, which has funded the chirality research in our lab for the whole time I've been a professor. Well, not every year, but most of the time. And if you want to learn more about this, Tim mentioned I'm organizing with Tom Wenzel a conference in Boston about all aspects of molecular chirality. But if you don't want to fly east, you could fly west and go to Hawaii, where we will have a huge symposium on chirality that I'm organizing, covering different aspects, especially applications. So finally, I would like to thank you for coming and listening to my talk. It's been a pleasure to be with you this week, and I'll be glad to take any questions you may have.
You said that chirality is the phenomenon that is actually found, that is based on forms of life that are based on carbon.
Yes.
So are you trying to suggest that carbon is the only element that actually shows chiralic activity?
No, no. I think all I can say is that if the life is based on carbon, then we should see chirality in the molecules that are involved in the life. But you're right, there could be other forms of life that are not carbon-based, and then finding asymmetry in carbon wouldn't help to find those forms of life.
No, but because I think Nobel Prize for Chemistry was awarded to a chemist who proved that nitrogen also shows chiral activity, so that's why that question came up.
I'm not remembering that Nobel Prize, but nitrogen can show optical activity if it is bonded to 4 different things. It's usually only bonded to 3, in which case it doesn't show the chirality. But your question is still valid. There are other elements that may form the basis of life in ways that we simply haven't imagined. And they may or may not be elements that would show chirality. If they are atoms that would have the property, that normally have the property of chirality, then we should see chirality if there's life. And if there's molecules recognizing each other, it's back to that handshake. They have to both be right-handed or both left-handed for the recognition to work. So, for example, phosphorus would be another atom that is chiral with either 3 or 4 things attached. And there are many other possibilities, but that's a great question.
Is there any evidence that microbes use properties of chirality to evade host response?
To invade? Evade. Yes, absolutely. In fact, there's a famous drug, vancomycin, that, and I'm losing the details at the moment, but in bacteria, in the cell wall, you have D-alanine sequence. And I don't remember how it works. Tim probably knows, but anyway, the bacteria does use, in some cases, the mirror image amino acid, and it can be to its advantage because, our enzymes would recognize only the L amino acid. So it's a very insightful question. And I'm forgetting how this drug works, but it probably doesn't matter because now the bacteria have found a way to resist it anyway. But you are correct that can and it does occur.
In the example that you were talking about the propeller-like structures or the alpha helix and the beta sheet switching based on the addition or subtraction of an electron, is either in the lab or in nature, is that a simple application of current, or is there a more complicated system involved in that?
We have done it with simple current in the lab. There certainly are many examples of protein conformational changes with the redox change. I'm not aware of a protein that does the helix to sheet conversion itself. But yes, in the lab, current will do this. We have used current to make the switch. So the sheet structure then aggregates into larger structures typically. And so you can imagine being able to control materials in this way to aggregate and deaggregate peptides.